A warm greetings to all. This slide is about mitochondria, powerhouse of the cell. The term mitochondria is derived from the Greek words mitos and chondrion, which means thread and granules like. It was first described by German pathologist named Richard Hallman in 1890. This is the diagrammatic representation of mitochondria. Occurrence and distribution. Mitochondria are present in all animals and plant cells. They are absent in mature mammalian erythrocytes and bacterial cells. Mitochondria are found throughout the cytoplasm, distributed evenly, but there are many exceptions to this rule. In leukocytes, they are radially arranged. Muscles lie in rows sandwiched between contractile elements. Tailed sperms found in middle piece forming mitochondrial spiral. During mitosis, they are concentrated near the spindle. Movement. Under a phase contrast microscope, mitochondria are seen in living cells, stained with the vital dye Janus green B, which, though not entirely harmless, permits the cell to remain alive for a long time so that can absorb the mitochondria in motion. Under dark ground illumination, they are seen in the state of movement, which is of two types: transposition, wriggling. Movement of mitochondria is less in animals than in plants. Morphology, shape. The shape of mitochondria varies from cell to cell, but more or less constant in cells of similar types and cells having similar function. The mitochondria may be filamentous, spherical, rod shaped, granular, vesicular, or thread like. They may swell out at one end to become club shaped or hollow out at one end to assume a shape of a rat. And in a variety of other cells, highly branched mitochondria are seen. Shape may be changed 15 to 20 times in 10 minutes by heat, hypertonic and hypotonic media, by acid, etc. In acid pH, they tend to become vesicular. Size. After the nucleus, mitochondria is the largest organelle in the most plant cells. The size of mitochondria also differs from one cell to another depending upon the functioning of the related cells. They contain the diameter from 0.2 to 2 mu, and they are as long as 20 to 40 as in the developing oocytes of amphibia. They may therefore be similar in size to bacterial cells. Number. The number of mitochondria present in cells depend upon its metabolic activity. Cells with high metabolic activity have them in greater than present in cells with low metabolic activity. The cells of green plants contain less number of mitochondria in comparison to animal cells, because in plants the function of mitochondria is taken over by chloroplast. Typically, a cells may have 50 to 5000 mitochondria. Structure. The mitochondria is a double membraned rod shaped structure found in both plants and animal cells. Its size ranges from 0.5 to 1 micrometer in diameter. The structure comprises an outer membrane, an inner membrane, and a gel like material called matrix. The outer membrane and the inner membrane are made up of proteins and the phospholipids layers separated by the inner membrane space. The outer membrane covers the space of the mitochondria and has a large number of special proteins called porins. Cristae. The inner membrane of the mitochondria is rather complex in structure. It has many folds that form a layered structure called cristae, and this helps in increasing the surface area inside the organelle. The cristae and the proteins of the inner membrane are in the production of ATP molecules. The inner membrane is strictly permeable only to oxygen and ATP molecules. Several chemical reactions takes place within the inner membrane of mitochondria. Mitochondrial matrix. The mitochondrial matrix is a viscous fluid that contains a mixture of enzymes and proteins. It also comprises ribosomes, inorganic ions, mitochondrial DNA, nucleotide cofactors, and organic molecules. The enzymes present in the matrix plays an important role in the synthesis of ATP molecules. The matrix space contains all the enzymes and cofactors involved in Krebs cycles. Particles. The F1 particles are also known as oxysomes or elementary particles or F1 particles. These particles are present in matrix side of mitochondrial inner membrane. The F1 particles functions in synthesis of ATP. F1 particles comprises about 15% of the total inner membrane protein. Function. The main function of the mitochondria is to produce energy through the process of oxidative phosphorylation. It is also involved in the following processes: regulates the metabolic activity of the cell, promotes the growth of new cells and cell multiplication, helps in deoxifying ammonia in the liver cells, play an important role in apoptosis or programmed cell death, responsible for building certain parts of the blood and various hormones like testosterone and estrogen, helps in maintaining an adequate concentration of calcium ions within the compartments of the cell. It is also involved in various cellular activities like cellular differentiation, cell signaling, cell senescence, controlling the cell cycle, and also in cell growth. Thank you for watching.